Welcome to the second part of the Chapter 8 video lecture. Today we're going to go through everything after that first part of the chapter that was review of DNA structure and function, gene expression, so transcription and translation. Now we're going to move on and look a little more closely at bacterial genomes. All at that point, our genetics are the same, whether we're bacteria or a human. We've got nucleic acids as our building blocks, and we go through replication in a similar manner, other than we have thread-like chromosomes rather than a single circular chromosome. So now we'll look in specifically at our bacteria. In section 8.3, we're looking at operons. So we're going to talk about the lac operon. And the lac operon, lac stands for lactose. Operons are the way bacteria regulate whether or not a gene will be expressed. So when we talk about gene expression, we mean the making of a protein. One gene, one protein. We don't always want to make every protein. It takes a lot of energy to make proteins. We don't always express all of our genes all the time. We use that analogy of a cookbook to a recipe to an outcome. We used, the whole cookbook, a recipe for a cake. The outcome is the cake. It's the equivalent of our DNA is used to transcribe to messenger RNA, is translated into a protein. We don't always want or need every protein. Those proteins are enzymes with a job to do. So when we want them to do their job, we want them. But when we don't need them, we don't want to use the energy and the resources to build them. A really good analogy of the operon system and why bacteria need a way to regulate expression of their genes would be similar to your phone and all the apps on it. If we think of the genes as the apps on the phone, you don't want all your apps running all the time. Why not? Well, it would eat up resources, your data, your battery. We only want the apps on when we're using them. Similarly, bacteria, all cells, they don't want to make all their genes, all their proteins all the time, only when they need them. Eukaryotes have some different ways to regulate them, but in bacteria, they use the operon system. What the operon system is it's a way to regulate whether or not our DNA is expressed through this length of DNA before the gene. So the gene, one protein, the genes code for proteins. And we want to regulate that expression. So along the DNA, right before the actual structural proteins for the enzyme that I want, is a section with control pieces, ways to manage whether or not that gene gets expressed. Remember, gene expression means transcription and translation. So how do I keep that from happening or make sure it happens? We can have different types of genes. So let's see here. We're looking at a gene. This is a part of the chromosome. This is one stretch along a chromosome. So this is my DNA, right? My DNA has the information. Each locus is the site with information for how to build a protein. One gene, one protein. I have some genes that I always need that protein. Those are called constitutive genes. They're always on. I'm always making that protein. I'm always making that enzyme. So things that the cell needs all the time. I constantly need to replenish parts of my cell membrane. So I'm constantly making the enzymes I need to make those phospholipids, to make the proteins that belong in the plasma membrane, to replenish those. So those are constitutive genes. There are some genes that I want on until I no longer need them, and then I want to be able to turn them off. These are called repressible genes. Repressible genes are on building their enzymes all the time until something turns them off. Typically, repressible genes are involved in anabolic chemical reactions. Anabolic reactions build big molecules from smaller ones. So if I'm building big molecules from smaller ones, I need a bigger complex molecule. I need to build it. I'm going to keep making those enzymes until I don't need them anymore. What's going to indicate I don't need them? Well, when I have enough of that big molecule at a certain concentration, I'm going to say, okay, that's plenty, let's stop. So the presence of that big molecule at a certain concentration will turn that gene off. It can be repressed. Inducible operons, like the lac operon, are typically catabolic. They're involved in breaking big molecules down to small ones. I don't need the enzymes that do that if that big molecule is not present. So if lactose is present and I need to break it down in order to metabolize it to get ATP from it, then I'm going to need to make those enzymes. Usually glucose is my preferred energy source. It's the most abundant sugar in the world. And so that's usually what I need, the enzymes for that. But if all of a sudden I don't really have glucose or there's a lot of lactose, that's much easier to obtain. Well, now I need to turn that app on. I need to make those enzymes. So here's my operon. And the operon includes all these different components. It includes the system that actually regulates whether or not I can transcribe and translate the genes I need. And it includes the structural genes or the operon genes. These structural genes these are the enzymes I want to make. So my operon genes are my structural genes. You'll see separated, and this is just to indicate that it's probably some distance along the DNA, along the chromosome, before I get to the actual genes, to the actual operon. The regulator, remember this is DNA, so each section is a gene. Oops. So this gene here, my regulator, or my regulatory gene, is going to code for a protein, one gene, one protein. The protein that it makes is this molecule here. This is my repressor, or my repressor protein. This is an inducible gene, meaning it's usually off or it's usually repressed. It's usually prevented from making these enzymes. So the repressor protein's job is to stop transcription. If I don't get the messenger RNA, then I can't get the protein through translation. So this regulatory gene is somewhere close to my actual operon, but not necessarily attached to it. It could be somewhere before the actual operon. It might be attached to it, but usually they're found before, and there's some other DNA in here. This doesn't indicate a hole in my DNA, in my chromosome. It's just not showing it to show that there's separate sites. My regulatory gene gets expressed through transcription and then translation to make this protein. This protein, or protein shape, is specific to its function. The specific shape of this allows it to sit down on this segment of DNA. This is called the operator. The operator works like an on-off switch. If the repressor is there, my call can't go through. Operator, put a call through. If that's blocked, the call that can't go through is RNA polymerase. Remember RNA polymerase, ACE, the enzyme that builds the polymer RNA. That's transcription, building RNA, my messenger RNA from my DNA. RNA polymerase knows where to start transcribing because this promoter. The promoter acts like a flag. It indicates to RNA polymerase, hey, come in here, start here. This is where you want to start transcribing. So RNA polymerase lands on the promoter, and notice that promoter is separated from the genes. That's because of the regulatory function here. Yep, this is where I want to start, but now to continue on down this section of DNA in order to transcribe it to mRNA, I can land here, but as I move forward, the operator is what tells me, yes, you can go ahead, connect the call, or no, you can't. I'm going to block it. If the repressor is present, the operator blocks the RNA polymerase. It's a physical block. The RNA polymerase can't proceed and can't transcribe these genes. So I've got my promoter, which is where my RNA polymerase is going to say, oh, here's where I start transcription. Let me see if the operator will let me. Oh, no, the operator is shut down. I'm locked out. No transcription. So this is typically off. It's inducible. It's inducible by the presence of an inducer molecule. Well, that's the molecule lactose. If lactose is present, then I want to transcribe these genes into messenger RNA so that I can translate them into a protein. Notice, again, our protein shape. This part's the exact shape to let it land on the operator. This part's the exact shape of lactose. If lactose is not present, this molecule sits here. Lactose is my inducer. It can induce this gene to turn on. When it lands in the repressor protein, it attaches here at this active site, and it makes a change to the shape of my repressor. Once that lactose is attached, the shape changes and it falls off the operator. Now my RNA polymerase is free to proceed down through those structural or operon genes and transcribe them into my messenger RNA, which will make its way to the ribosomes, which will then allow transcription or translation to take place. And I will make my 3 enzymes needed to break down lactose and get energy out of it. These enzymes will work until there's no substrate. So lactose is the substrate they act on. They convert it, we get ATP out of it. And they'll keep going and keep working, and I'll keep making these enzymes until all the lactose is gone. The last bits of lactose will fall out of my repressor, be metabolized, and then my repressor will change back to its original shape and land on the operator and block further transcription. So that's how my operating system works. That's its purpose, is to help my cell to be energy efficient by only expressing the genes I need only when I need them. So let's see what we got. The lac operon is always on when glucose levels are high and lactose levels are low, on when glucose levels are low and sucrose levels are high, on when glucose levels are low and lactose levels are high, or always off. The lac operon, the enzymes I need to break down lactose, are only going to be on when lactose is present at high concentration and when my glucose is low. If glucose, as long as there's glucose, I'm going to use that. That's the easier one to use. So glucose has to be low and lactose has to be high before I switch my lac operon on. So remember, the lac operon, it is inducible. It is usually off, but the presence of lactose indicates that, hey, I need to turn it on so that I can use this lactose for some energy. All right, on to the next section. We'll look at the types of recombination pretty briefly. Recombination is important in bacteria. This is our question in the discussion some people are struggling with. In eukaryotes and in organisms that can sexually reproduce, we get half our genes from mom, half from dad. That means every time I get a new offspring, I have a different combination of genes. That's because when dad goes through meiosis to make sperm, we shuffle his genes around and half of them end up in the sperm. And when mom makes the eggs, we go through meiosis, we shuffle the genes around and get half of them. Well, in that process, we never know which half we're going to get. And then fertilization, we bring these 2 different mixes of chromosomes from mom and dad together to get our diploid, 46 chromosomes in humans. And We get new combinations every time. So unless you're a monozygotic twin, an identical twin, there is nobody who has the exact same set of genes as you. You may share some with your siblings, you share some with your parents, but the grand total of all the genes are different in every individual. So bacteria, though, they reproduce through binary fission. It's cloning. They make an exact copy of their DNA in DNA replication, and then they split those two copies. So they start with one, they end up with two. They split those two copies between daughter cells. Those daughters are identical. They're clones. And every time we go through binary fission, we get more clones, with the exception of random mutations that only get through about 1 in every 10 billion base pairs copied. That's because our DNA polymerase is such a good proofreader, and because complementary base pairing really minimizes the number of mistakes I make to begin with. So bacteria, to have variation in their population, in their genetics, and in their phenotypes, have come up with different ways to get variation. Variation is really the basis for evolution. Populations are more fit if there are more varieties of phenotypes and genotypes. Natural selection acts on the phenotypes. Some phenotypes are more fit than others, so they'll reproduce at a higher rate, pass on their good genes at a higher rate. But if the environment changes, it might be a different phenotype that's favored. Because of meiosis, because of sexual reproduction, we have lots of variety. Every round of reproduction, we get variety. Every offspring, everyone is different. That's a lot of variation. And some varieties are more fit than others. Bacterial clones, because of binary fission, if something was harmful to one bacteria in the population, it would be harmful to all of them unless variation arose. Mutation at that background rate's just not high enough to confer a real benefit, so bacteria have evolved ways to get that variation by sharing genes with other bacteria, not just of the same species, but of different species. So here are the ways they do that they can recombine and reshuffle their genes. 3 of these are horizontal gene transfer. Horizontal gene transfer is when bacteria exchange pieces of their genome with other bacteria, not passing it parent to offspring, but kind of within the same cohort, not necessarily in the same generation, but it's not parent offspring. It's some other bacteria around me. Typically, we're going to exchange plasmids, just small pieces or chromosomal fragments, not the entire genome, just small pieces. And those small pieces can and usually do carry some sort of beneficial trait. We're not going to transfer an entire genome. Every bacteria already has its own genome. That's what it uses for survival. That has the information for how it needs to function. So these are just like bonus pieces of DNA. The 3 types of horizontal gene transfer involved in recombination are conjugation. Don't confuse this with sexual reproduction. Our bacteria never have mom and dad coming together and sharing half of their DNA to get new combinations. Conjugation is so named because the 2 bacteria move close together. And then when they move close together, they're going to exchange a piece of DNA, a plasmid, between the donor and the recipient. They are both living cells, live functioning cells, and sometimes it's called a conjugation. Conjugation, we refer to that pilus as the conjugation pilus. So usually that's how we see it. That's how it was first described, was with a pilus, and we thought that was the only way this could happen. So the pilus brings the 2 bacteria close together, and then they exchange a piece of the chromosome, a plasmid, that usually confers some benefit. Drug resistance, toxin resistance are some of the more common plasmids shared through conjugation. Transformation. When organisms die, They decompose, but they don't just decompose all at once, poof, into schnapps, my carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. They break down little by little. And part of that breakdown can leave behind small fragments or pieces of DNA. And bacteria are able to actually bring them in through their cell wall, through their plasma membrane, and incorporate them into their own DNA. Sometimes that's beneficial. And then transduction is when pieces of bacterial DNA are transferred to other bacteria through an intermediate, through a bacteriophage, which is a virus that affects or attacks bacteria. And when they go in, they release their nucleic acid in, and it incorporates in with the bacterial DNA, and then forms new viruses. We're going to learn all about viruses and how this happens in chapter 5. But when it forms new viruses, sometimes it doesn't just incorporate the bacteriophage DNA into the new viral particle, but it will take pieces of the bacteria's DNA. And then when it infects another bacteria, it will incorporate that piece of DNA into the new bacteria. So it will take a piece out with its own DNA and move it to another bacteria. Sometimes that is just random. Whatever segment of DNA it happens to be close to, it carries with it. Sometimes it's very specific. The viral DNA is a very specific match through that complementary base pairing with a specific segment of the bacteria's DNA. And so it will pull out a specific part of DNA, a specific gene. So we call that specialized transduction. When it just takes random pieces, that's generalized transduction. The fourth way that bacteria can change their genome is through transposons. You'll see these referred to as jumping genes. A transposon is a section of DNA in a bacteria that's able to replicate itself and then move to somewhere else in the genome. Sometimes it'll flip itself upside down when it does that. Depending on where it inserts itself, it might insert itself into the middle of another gene and cause a new protein that's never been seen before to be made. This could have detrimental, lethal effects if it interrupts production of a necessary protein, or it might come up with some novel protein that makes it more fit. It can also jump out of the chromosome and into a plasmid and be carried on to other bacteria. So transposons allow for some more genetic variation in bacteria. And all of those allow bacteria to have more variation in their population, which allows the population, not necessarily the individual bacteria, but the population over time will be more fit. All right, concept check. Which of the following mechanisms of horizontal gene transfer involves the transfer of bacterial DNA through a bacteriophage? A virus that affects bacteria. Is that transposition, conjugation, transformation, transduction, or none of the above? Yay, transduction. Right. Okay, our last section that we're going to go over is on mutation. So we'll look at some of the mutations that can occur, and then we'll look at activity 1 and the question on mutation to help you with that. A mutation is a change in the genetic code. Changes are our mutation, our change to the genome is the driving force behind evolution. So any change in the nucleotide sequence is a mutation. This is most noticeable if there's a change in phenotype, but it could just be a change in genotype. It can involve the loss of base pairs in our nucleotides, the addition or the rearrangement or the order of base pairs. So any change to that nucleotide sequence is a mutation. So mutations arise randomly, spontaneous mutation. They're just random changes in the DNA due to an error during replication. As we saw, DNA polymerase is a really good proofreader, so it doesn't happen very often. When it does, we can have lethal mutations, some change that leads to an error in building a critical protein, and so the organism can't survive without it. Those will not be passed on. We can have a neutral mutation. It really doesn't make any change in the fitness of the organism, or it may slightly decrease fitness, but it doesn't help the organism. So it stays in the population. As that organism undergoes binary fission, it passes on that mutation to its offspring, but all the other thousands of bacteria in that population are passing on their unmutated DNA. So it's there, but just at a really low level. Usually, a mutation is not going to confer a noticeable benefit initially. When we know if a mutation is beneficial or not is if the environment changes. So let's say a protein on the cell surface mutated, and that protein was an entry point for an antibiotic. If that mutation changes the function of that protein has to have some other function. There's not a protein that just allows antibiotic in. It allows something in, and the antibiotic is targeted to take advantage of that. So if that protein changes, well, it won't be able to do its real job, or maybe just can't do it as well. So the bacteria might be a little deficient, but it hasn't killed it. So it's passing it on. So all its offspring have that change. And now all of a sudden, we introduce an antibiotic. All the bacteria without the mutation die. This bacteria, because that surface protein is a slightly different shape, because it had a different amino acid, the antibiotic can't get in, and that bacteria survives. All its offspring survive. Yay! And now that bacterial population is resistant to that antibiotic. So a lot of times we think about induced mutations. These are those that result from exposure to known mutagens. Known mutagens cause random changes or disruptions in the DNA. We can't control UV light where that change is going to take place, where in the DNA, or X-rays. They're going to cause mutations. They cause mutations to occur at a higher rate, but they're random mutations. So exposure to an antibiotic isn't a mutagenic agent. It doesn't cause mutations that confer resistance. The resistance has to be present before it's beneficial. Because if it's not present, antibiotics kill bacteria, it would kill them. So when we talk about mutagenic agents, we don't induce specific mutations that give a benefit to that mutagenic agent. So if that was true, then radiation would make organisms resistant to getting cancer from UV light. That doesn't happen. We get cancer because we get a mutation from it. So the mutation is random, or it can be driven, but where it occurs and what effect it has is random. It's not controllable. The mutation, though, to be beneficial, already has to be present in the population. So positive and negative effects, they're permanent and inheritable. Most are not beneficial, but if the environment changes and that change to the genome and phenotype, to the phenotype confers some benefit, then it will help the population to adapt. As long as the environment's stable, mutants will just make up a small portion of the population because they don't have a benefit compared to the other organisms. So we'll talk about that more when we talk about antibiotics and drug resistance, but just know for now that mutation has to be present first. The antibiotic doesn't drive the mutation. The mutation has to be in the population. Otherwise, the antibiotic will do its job and kill all of them. All right, so some types of mutations. Here is my normal protein. That's my template strand that codes for this messenger RNA in transcription that is then translated into these amino acids. And I use my codon table to figure out what each of these codes for. I can have a missense mutation that leads to an amino acid in the chain being switched. So a missense mutation, we talked about sickle cell anemia. It's an example in your activity. I have a switched amino acid. So here, by changing my G to a T, that changes my codon to GAA instead of GAC. And instead of aspartic acid, I think I get glutamate. GLU. Instead of ASP, I have GLU. I have one changed amino acid. This is going to probably change the shape and function of this protein. So it's probably not going to function as well. It may or may not be lethal. As we saw with sickle cell anemia, it has a lot of adverse effects, but not lethal. So that's a missense mutation. I end up with the wrong amino acid because a base was changed. I can have a base substitution that results in a silent mutation. So here, if I change that G to an A, I change my codon from GAC to GAU. Go to the codon table and look, because the genetic code is redundant, multiple codons code for the same amino acid, I have no change. So a silent mutation. Here I have frame shift mutations. Remember, my reading frame is these three nucleotides or nitrogenous bases, these three. There's no overlap, so I don't read AUG, UGA, GAC. I read that triplet, that triplet, that triplet. Here, if I delete this G, what happens is I still read as triplets. So I read TAC, TGC, TGC, TCT. I've shifted my reading frame. Now, when I transcribe that, I have all these different base pairs here. I have a frame shift in my reading frame that results in a whole bunch of different, these are still the same, and then I get, wrong amino acid, wrong amino acid. Here, that shift in reading frame puts a stop. I'm not going to get a protein. I'm going to get a polypeptide, not even poly. Poly's many. This is here a couple. 1, 2, 3, 4. 4 amino acids. They're not going to be able to fold into the right shape. They're not going to be able to do their job. This protein is not going to get made. Protein synthesis stops right there, and I don't get that protein. This is called a nonsense mutation. Because I'm not getting a protein that I need, this is going to be lethal. I could also, depending on where this frame shift mutation happens, let's see here, I insert an amino acid or a nitrogenous base in my DNA. I get GCT by putting that guanine in here. I shift my reading frame. Here, I don't shift to get a stop. I just shift, but I have all different amino acids. So this can go on until I get to a stop codon. But I'm going to have the incorrect amino acid order, which means whatever that folds into is going to be wrong. So it'll probably be lethal. So those are my different types. I can have a missense mutation, just get the wrong amino acid. A silent mutation, the change, but I still have the same amino acids. A nonsense mutation where I get a premature stop codon, so I don't get the protein. Or a frame shift mutation that is just going to give me the wrong protein altogether. So looking at number 10 on our activity, it gives part of the chain of my polypeptide for a normal hemoglobin molecule and for a sickle cell hemoglobin molecule. And we see the amino acids are the same for valine, histidine, leucine, threonine, proline. But right there where I should have glutamate, in sickle cell I have valine. This is GLU in your codon table. This is VAL. And the question is, what's the minimum change necessary in the mRNA, in the codon, to get that change? So what you'll need to do is go to your codon table, look up all the possible codons for glutamate, look up all the possible codons for valine, and figure out what's the smallest change needed to end up with sickle cell. All right, last concept check. Which of the following types of mutations will have the most devastating effect on a cell? A point mutation, or just change one of my nitrogenous bases. A frame shift mutation, where I shift my entire reading frame because I've either left out, skipped, or lost a nitrogenous base or added one. A missense mutation, where I get the wrong amino acid in a specific place. A nonsense mutation, where I get a premature stop codon. Or a silent mutation, where even though my DNA changed, my codons, because of that wobble in my codons, because of redundancy in the genetic code, I still get the same amino acid. So the answer really should be a nonsense mutation, where I get a stop codon. I don't get that protein at all, so whatever its role is doesn't get done. But a frame shift mutation, because it can either lead to the wrong protein, so that job won't get done, or an early stop, a nonsense mutation, can have the same effect. So I would say both of those. I would take either one of those. All right, well, I hope that helps get you through activity 1, and post any questions you may have in the discussion. Thank you.